Hi, my name is Genís. I'm a psychologist and pharmacologist, and I will present you the main findings of a recently published systematic review about the use of cannabis sativa for pest control. Well, first of all, I'm sorry for my English. I'm talking from Spain. Maybe some words sound awkward for you, but I hope you can understand me. And well, it is a pleasure to give you this lecture because I think Oregon State University is one of the main hubs of hemp research. So it is a very source of inspiration for us. And well, just for provide you with some context of this research was performed while I was doing a doctoral stay in the Botanical Institute of Barcelona. This institution has been working with different projects about cannabis. For instance, they published this database about the traditional uses of cannabis, can use. And well, they have been performing other projects. In addition, I'm working in ICERS, in this NGO, which is working in the intersection between policy, human rights, and research regarding the traditional use of psychoactive plants, including cannabis, of course. And my supervisor in the university has a lot of experience researching with pesticides, basically chlorpyrifos, and its consequences to health exposure. So, as you know, the research was making a lot of sense given the institutions involved. No? So, well, the first question is why cannabis, right? The use of chemical pesticides has been increasing in last years due to intensive agriculture, as you know. And we are aware of the potential negative consequences of the exposure of these chemical pesticides. And this increased the interest for greener and eco-friendly alternatives to these pesticides. No? So cannabis, in this regard, is a very good candidate since it is a plant that it is very rich in essential oil, so the production of essential oil is very efficient. It is a complex product containing more than 500 compounds. So in terms of its phytochemical profile, it's very rich. And it is a plant with different uses. We can use the plant for the extraction of the essential oil, but we can also use the plant for fibers or for food and for other uses. No? And well, this research was performed in two different parts, two different reviews. First of all, we performed a first review about the evidences of ethnobotanical uses of cannabis for pest control in this database, which was developed by the team of the Botanical Institute, as I said. And then we performed this systematic review about the experimental studies which assessed the efficacy or the toxicity of cannabis sativa against different pest organisms. So well, the first search in this database retrieved 9 different references. As you can see, most of the references came from Asian countries, and most of them used leaves of the plant for the juice or for the extracts that finally were used. And the conclusion of this first search is, well, we can summarize it saying that it is a well-established use of cannabis sativa for pest control in some countries, mainly Asian countries. And the second part of the search consisted in this systematic review, for which we used these databases. And well, the first search retrieved nearly 600 records, selecting 30 final references for this review, for analyzing in this review. And well, the results here are a bit more complex because most of organisms that were tested consisted in Arthropoda phylum, mainly Insecta and Arachnida. And I should note that all the Arachnida organisms that were tested against cannabis showed a very high toxicity, were highly affected by cannabis, including these 2 ticks. And in the case of insects, they were also highly affected for cannabis. And 17 different species of insects were highly affected by cannabis, including this Aedes aegypti, which is a vector of different viruses, as you know, and this small beetle with quite an unpronounceable name. But there were also some insects that were not affected by cannabis, like these ones, Reticulitermes virginicus and Drosophila melanogaster, which is the common fruit fly. But, well, most of the insects were affected by cannabis, as I said. And, well, other organisms were tested as well, for example, nematoda. We can see that the first 3 nematoda that you have at the top were highly affected from cannabis, while the 3 nematoda at the bottom were not affected by cannabis. And interestingly here, wild hemp was used for the extraction of the essential oil in the case of the nematoda that were not affected by cannabis. And different chemovars and cultivars were used for the essential oil in the case of nematoda that were affected. No? So this is suggesting that wild hemp could not be as effective as specific chemovars or cultivars of cannabis. The single organism belonging to gastropoda were also highly affected from cannabis. And different species of fungi were also tested against cannabis. And Sclerotium rolfsi and this Cryptococcus were also highly affected from cannabis. So we can see a high toxicity against different organisms. And, well, just for discussing some characteristics of the studies included, we can note that 17 out of 30 studies used wild hemp for the extraction of the essential oil, while the others used specific chemovars of cannabis. And as you can see, we can expect very differing phytochemical profiles from wild cannabis, you know. So future studies should use specific chemovars of cannabis in order to standardize the phytochemical profile. No? Also, 15 out of 30 studies did not perform this phytochemical analysis. So we don't know which compounds are exerting pesticide effects of cannabis in 50% of the cases. And in the studies that did perform this phytochemical analysis, the most commonly reported compounds were these terpenes. As you know, they are highly reported in cannabis. And, well, other sources of heterogeneity in these studies, in the studies reviewed, were regarding the plant parts used, for example. Most of studies used leaves of the plant for the extraction of essential oil. 5 studies used inflorescences, and the rest of studies used different leaves or inflorescences, or also other parts of the plant, like roots or others. No? And this is relevant since we know that the highest concentration of different compounds of cannabis, including cannabinoids and terpenoids or flavonoids, are located in the upper inflorescences of the plant. No? So it is very relevant which parts of the plant we select for the extraction of this essential oil. No? Also, there were also different extraction methods reported. The most commonly reported were steam and hydrodistillation, but also other methods were found. And in this case, let me say that the works performed by your professor, the Dr. Selchaskov, are very relevant in that sense and should be taken into account when considering to standardize the procedures of the extraction of essential oil for future studies. And, well, some interesting findings that you can see is that, well, some of the studies reviewed, this one in particular, compared the essential oil extracted from leaves and from inflorescences. And it was found that the essential oil extracted from these inflorescences was higher toxic, exerted a higher toxicity against pest organisms. Here you can see the LC values, how they are much lower in the case of the essential oil extracted from inflorescences, in line with the previously discussed. And interestingly, in this study, the essential oil from cannabis sativa was exerting a higher toxicity than the essential oils from these two plants, which are currently considered very promising essential oils in terms of pest control. So this is very promising results. And another interesting finding is that some studies compared the efficacy of the essential oil in terms of if the essential oil was extracted from fresh material, from the fresh plant, or from the dried plant, So it was observed a general pattern in which the essential oils extracted from fresh plants were more effective than the essential oils extracted from dried plants. And this is making sense since we know that terpenes and other compounds are very volatile compounds. So when the plant is dried, most of these compounds are already gone. So the richness, the chemical richness of the final product would be affected by the dried process. And in the same study, let me say that it was used DET as a positive control. And it was observed that one of these essential oils extracted from fresh material exerted a higher toxicity than DET. So this is very promising again. And we have another study in the studies reviewed that used another positive control, in this case chlorpyrifos. And it was found in that case that chlorpyrifos was more toxic than wild cannabis sativa from Nepal against the species tested. And this is also encouraging for future studies to use more positive controls because, as you know, as you can see, we have only 2 studies that used these positive controls among the all studies reviewed. So the data at this time is very limited, and we need more information in order to make effective comparisons, in order to establish the current toxicity profile of cannabis. Interestingly, it is also important to know the toxicity of cannabis sativa essential oil against non-target organisms, about the organisms that are not relevant for pest control, that are not considered as pests, actually. And it was found that 2 studies tested different non-target organisms. And it was found that Harmonia xyridis and Eisenia fetida were not affected by cannabis products, while Cleon dipterum was affected by cannabis. So future studies, again, should test cannabis sativa products against different organisms in order to know its toxicity against other organisms, right? And well, regarding the potential mechanisms, first thing to note is that insects do not have cannabinoid receptors. This is very uncommon in the animal realm because most of animals have these cannabinoid receptors, but insects don't. And it is very relevant in that sense. And it could be thought that maybe cannabinoid receptors are exerting some role in the pesticide effects of cannabis. But in this case, as I said, cannabis sativa resulted in highly toxic effects for 17 different species of insects. So we should not conceive the cannabinoid receptors as a potential target involved in the pesticide effects of cannabis. Instead, cannabinoids could exert these pesticide effects through other mechanisms. For example, they can inhibit the acetylcholinesterase, which is leading to cholinergic poisoning because of high amounts of acetylcholine in organisms. And this is a mechanism well known in different pesticides like organophosphates or neonicotinoids. So this is a well-established mechanism. But additionally, cannabinoids can also inhibit P-glycoprotein. This glycoprotein, also known as PGP, is highly involved in multi-drug tolerance. Just for your understanding, when animals in general, including humans, develop tolerance to some drug, it's because more PGP is recruited, and this PGP is eliminating that drug. No? It is involved in the drug efflux of the cell. So it is actually involved in this tolerance. When you have more PGP in your brain or other tissues, you require higher doses of the drug to reach the same effects. So this is when tolerance happens. And PGP is facilitating this process as a critical compound. No? So it is kind of dismantling the defenses of pest organisms in that sense, because inhibiting P-glycoprotein, the organisms will be more susceptible to the effects of other compounds of the cannabis plant, right? And the mechanisms of other compounds like terpenes or flavonoids, we can cite also the inhibition of acetylcholinesterase, leading to this cholinergic poisoning. But terpenes can also modulate GABA and octopamine receptors, leading to neurotoxic effects. And flavonoids, interestingly, they can modulate ecdysone receptor, which is associated with changes in the neural response to food of organisms, so leading to the eventual death. And it can also inhibit the PGP, again, and the glutathione S-transferases, which is involved also in the detoxifying process of insecticides. So here we have different mechanisms that are dismantling or disrupting the defenses of organisms and leading to an enhanced effect of other compounds, like, for example, terpenes. No? So we can expect that the pesticide effects of cannabis would be associated not only with isolated compounds, but also the combination of different compounds in the plant, no? Which is commonly known as entourage effect. In the case of entourage, as you know, it is a concept only known and used inside the cannabis research field. So I prefer to talk about interaction effects. And in this case, we can expect some similar effects to the synergistic kind of interaction, no? Where you have an enhanced effect as a combination of different compounds. And well, just some final remarks. It is clear that cannabis sativa has potential for being used as a pesticide, but of course, further studies should improve their methodologies in order to standardize the methods used. In my opinion, this is not something that we included in the manuscript, but in my opinion, I have to say that the full potential of cannabis sativa as a pesticide has not been yet unveiled, because most of studies, as we said, they used leaves rather than inflorescences for the extraction of essential oil, or it was used a high amount of wild hemp, while we know that it may be preferable to use specific chemobars or cultivars. So I think that if all studies reviewed used the current knowledge in that regard, I think the results would be even more remarkable. And well, another thing to comment is that given some putative mechanisms at work, and I think in this inhibition of PGP and glutathione transferases, it would be great, it would be a good idea to combine the essential oil of cannabis with the essential oil of other plants. A thing that is currently being done, actually, with other plants, but not in the case of cannabis. Since we can expect that this inhibition of PGP and glutathione transferases would enhance the biological effects of the compounds present in the essential oil that can fight against the pest organisms. And well, this is just the main topics that I wanted to talk about. And you can, of course, contact me for any doubt or commentary about the content of this lecture. But thank you again for your invitation, and thank you for your attention. and I hope we can see in other forums. Thank you so much.